Okay, let's try this again. All right, we're talking about protein structure here. And when we're talking about protein structure, we're talking about the way that the entire protein is going to look. And a protein's shape is going to be the biggest determinant of what the protein does and what it can interact with. By interact, I mean basically just stick to, like two puzzle pieces stick together. And yeah, so in a protein, there are all these different bonds, and every one of these bonds is a point where you can rotate. So you can rotate this half of the molecule, and you can rotate this one relative to each other. And these atoms are rotating all the time. And so, yeah, you'll have lots of different motion on every one of these amino acids at every bond position. And part of that motion allows the molecule to either get out of the way or to then be in a position to interact with the side chain of another protein molecule. And that's generally how it happens, how interactions are done. So usually there are interactions that only involve the backbone. So the backbone is shown in this sort of purplish gray box, whatever color that is, down here at the bottom. And there are other interactions that involve the side chains. There usually aren't that many side chain to backbone interactions as far as I know. But that's fine. So if you haven't learned your amino acids yet, this is another good little reminder because we're totally going to be going over this in the next class. So yeah, make sure you know your amino acids. So these are all the non-polar or hydrophobic amino acids. Glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Yeah, turn them into some kind of word. I don't know. It can be done. Here, your polar amino acids. Now look at each of these side chains, which is the thing in the green box at the top of each amino acid. Can you see the atoms in that side chain that make this polar? Go ahead, look for them a sec, and then... All right, you see them? Yeah, it's like Dora the Explorer, right? So it's oxygen right here, sulfur up here, and nitrogen, and oxygen, and nitrogen, and oxygen, and oxygen. Yeah, those are the atoms that are going to make a side chain polar. Compare that to the hydrophobic side chains over here, and you've got hydrocarbons. Carbon and hydrogen, sharing evenly carbon and hydrogen. You got a couple down here, tryptophan and methionine, where there's a little bit of there's one of those atoms that seems to be a little bit polar, but because the rest of the molecule is so overwhelmingly hydrophobic, we can say that overall it's going to be hydrophobic. That's the category we would tend to put it in. But yeah, the more you know, the more you see that it's kind of a sliding scale. Yeah, from very hydrophobic to not very hydrophobic, very polar to not very polar. Yes. So here's your acids, and you'll see that in the acid form, glutamic acid, they are going to have a little negative up at the top. And if you add an OH on the end of that, then that would be glutamate or aspartate. You'll see it written that way sometimes. I believe at neutral pH in solution, they are going to generally have a negative charge on them, if I remember correctly. These are your basic amino acids, and again, at neutral pH, if I remember correctly, these things are going to have a positive charge on them. Although I think histidine doesn't get its positive charge until you get the pH down around 5.5. I think that's about where it picks it up. But yeah, lysine and arginine are quite positive and are at physiological conditions, like normal conditions inside the body, those would be positively charged. But all three of these you can put in the positive category. Here's a nice little thing. If you are thinking of making flashcards, use this slide. Make your flashcards. I need you to know that, or rather, I need you, if I say histidine, you have to be able to say H and that it is basic. And if I say tryptophan, you have to be able to say W and hydrophobic. But if I say E, you have to be able to tell me that is glutamic acid and it's acidic. Just like that. Got it? Yeah, easy. It's a thing that you can learn. It's a thing that you have to learn. And we'll go right into why you have to learn it because we're going to start using this stuff. And week after week, we are going to use these amino acids over and over and over again. And yeah, that's why I need you to get good at these. Um, yeah, I don't know if or or you can take another class because there's a lot of things you can do in this world, and it doesn't have to be this one thing. But if you want to do this one thing, biology, then this is what you need to learn. Yeah. So there we go. All right. How do we make a protein? By joining amino acids, of course. And so here's two amino acids joined together. This looks like a methionine. This looks like a tyrosine. And it looks like we're going to join on a cysteine. And you can actually go back a couple of slides and you can find these amino acids and check my math, see if I'm right. I think I've got those right. The way we make a protein is we're going to take the N-terminus, which is like the engine of the train. It comes at the front. That's going to stay, so the N-terminus of the new amino acid is going to come and join on to the C-terminus, the carboxyl terminus over here, the C double bond O, OH. And that's going to be like the caboose of our train. So N-terminus is the engine, C-terminus is the caboose, N-terminus is the beginning, C-terminus is the end, amino-terminus is the beginning, carboxyl terminus is the end. Those are different ways of saying the same thing. And whichever one works for you, good. But make sure you write that down so you got it. Because, again, I'm going to assume that if I have put it in these lectures, that you have got it. And if you don't have it, then things are not going to make sense down the road. All right. So the way we actually make this is it's called a dehydration reaction. We're going to take out an OH, hydroxyl, from this side, and an H, a hydrogen, from this side. And we are going to put those together, and we're going to join up what's left. So the carbon's going to end up joined to the nitrogen, just like so. And the H2O is going to go away. So we are going to create a water molecule from little pieces that we used. And in the process, we're going to join together another amino acid joining onto the end. And so you can see our new amino acid joins onto the C-terminus, and now it's the C-terminus. It's like a conga line, maybe. You can only join the end. Don't be a psycho and join the beginning of a conga line, or like push into the middle. Oh my gosh, yeah. What are you thinking? The society has rules. All right. So here are your ABCs of protein structure. There are three main kinds of structure that make up every protein. You've got an alpha helix, a beta strand, and a random coil. And these things are all made by hydrogen bonding together the backbone of the protein. So these are generally not going to involve the side chains, these groups that they're showing as an R, because the R is going to be the bit that is different on each amino acid. So you would think that, wow, I ought to be able to make an alpha helix or a beta sheet out of any old amino acid, but this is not the case. Because some of these side chains are better at getting out of the way than others, basically. Or can make additional interactions that will help stabilize the structure. So yeah, alpha helix, beta sheet, and random coil, ABC. All right, how do you recognize an alpha helix? So this is a thing that we are going to do, and we're going to have a little homework over this that you are going to do and turn in. So the homework is going to be, I'll show you that. I'll make another little video that shows that, but it's actually included right in the Blackboard site, right there. So there are amino acids M, A, L, E, and K. Now, do you know your single letter amino acid codes? If not, they are methionine, alanine, leucine, glutamic acid, and lysine, M-A-L-E-K. Those are the ones that are most likely to make a helix. You can remember this however you like. The way I remember it is that the word Malik comes from an old Babylonian or Akkadian, better, word that means king or queen, like Malik, Malika, Malkat, something like that. And so I'm imagining a king, King Malik, sitting on the throne. Maybe you know somebody who's got this name, Malik. Yeah, put them on the throne. And then instead of a crown, you're going to put a little helix on top. Boop, spiraling up there. It's a good-looking crown. That is a strong look for King Malik. Or, of course, you can remember this however the heck you want. But all I ask is that you definitely remember it. So that's going to be the useful bit here. The bit that you don't need as much, but in case you're interested in how things work, which I hope you are, if you have these amino acids, proline, tyrosine, glycine, and serine, they're going to have a low chance of forming a helix. So you're often going to have those in between helices or in some other type of structure. A proline actually puts a kink in the chain. So you'll often have those at the start of a helix, but not at the end, and usually not in the middle of a helix. And D and E tend to be helix breakers. They tend to occur at the end. So both of the acidic amino acids. And for beta strands, of course, there's another little set that are more common in beta strands than in alpha helices, like, significantly more common. And so these are going to be, let's see, threonine, valine, isoleucine, tryptophan, phenylalanine, and tyrosine. I guess I did those in the wrong order. But the way I remember it is TV Wi-Fi. So I imagine, I don't know, like a 200-year-old TV where you had to adjust the little antenna on the top of it, and the antenna looks like a little sheet. Or, I don't know, maybe imagine it's like a nice flat panel TV, because these make a nice sort of flat sheet. And so, yeah, this is my new Wi-Fi enabled TV, and you just have to remember to spell Wi-Fi wrong, W-I-F-Y. Or whatever. Remember it however you like. Yeah. You're not going to find prolines in a sheet, but they will often occur at the edge of a sheet, and they'll make a thing called a turn, because prolines, yeah, put a little turn into the molecule. Okay, all the other amino acids, these are things that can occur in a helix, can occur in a strand, can occur outside. It's just, yeah, generally you're not going to find them in there. In an alpha helix or a beta strand, they're not going to be predictive. I guess that's a better way of thinking of it. Our third type of structure is random coil, which can be hydrogen bonded. And each of those hydrogen bonds, if you're into physics, is going to have a free energy, rather, of, about -1 kilocalorie per mole. Yeah, so it's a pretty decent contribution. And something like a interaction between a virus and its receptor or an antibody and its target is going to be in the 10 to 20 kilocalories per mole range in terms of energy. So if you happen to see alanine, proline, glutamic acid, and serine, these tend to promote disorder. But the way I'm going to work this when we do quizzes and tests is I'm going to box an area, and you're going to tell me, does it have more amino acids that are like M, A, L, E, and K, or does it have more amino acids that are like T, V, W, I, F, and Y? And whichever one is the more common within that region is going to be the right answer. And the other thing we can do is we can stack proteins on top of each other. We can line them up so that the same parts are stacked on top of each other. And so then instead of just predicting for one protein, we can predict for all of these related proteins. Because if they're related, they will have the same structure. And so that can help you narrow down the options, basically. Yeah. All right, so what's the point of all this protein secondary structure? Well, we got to make a protein shape. Yeah, we got to make a tertiary structure for our protein. And every protein is going to have a surface. And so, look, this is the hemagglutinin protein from the influenza A virus. This is the flu virus that, people get sometimes every year. And you see the surface on this is a really good match for the surface on this. It sticks up here, but that's kind of at the back, and there's actually a little pocket here at the back. So this fits on shape-wise really well. Now, the antibody protein is a thing that your immune system will make to stick to and block these proteins from actually being able to work. And so, yeah, they're part of what keeps you healthy. So in addition to matching the shape, if two proteins are going to stick together, and proteins sticking together is kind of what goes on inside of a cell. This is how everything works. You have to not only match the shape, you have to also match the charge. So if it's polar up here, it needs to be polar down here. If it's hydrophobic up here, it needs to be hydrophobic. If it's acidic over here, it needs to be basic over here, or at least polar, but not hydrophobic. Definitely not. Because a polar will have a little positive side and a little negative side. It's just not as strong as having an actual +1 or -1 charge. Okay, so there's 4 levels that we can think of protein structure on. The first level is primary amino acid sequence. And so this is going to be those 20 single letter amino acid codes in a row, in a chain. And the first one's going to be the N terminus, the last one's going to be the C terminus, and that'll show you the order that they're joined up in. But that doesn't tell you much. And so what we've just learned is that you're going to be able to look at that primary amino acid sequence, the primary structure, and you're going to be able to see regions where it goes M, L, L, A, K, K, E, something like that, and say, whoa, that really looks like an alpha helix. Look at all those alpha helix type amino acids in a row. And you're probably going to be right. You'll be able to look at these, it looks like a chunk of letters, and you'll actually be able to see things and start to see how a protein works. That's the level where you kind of know that you've started to understand biology. And believe me, there's a ton more to understand beyond that. But if you can get to that level, yeah, things are going to be okay. But don't rush it. You don't have to get there right away. Let's see. So secondary structure is what we just learned about. This is the alpha helix, beta strand, or random coil. What does each amino acid do? And so there's going to be a bunch in a row that'll be an alpha helix, a bunch in a row that are going to be a beta strand. Fine. Third level, tertiary structure, is the three-dimensional shape and three-dimensional surface of the proteins. And so, yeah, that's going to be determined by secondary structure and the various side chains, how far they stick out, what charge they have, or if they're polar or hydrophobic. And then the fourth level structure is how different proteins will fit together, like little Legos or like little puzzle pieces, to make larger machines. Because just like with a box of Legos, the little bricks are okay, but it gets really interesting when we start to combine these together. And this happens a lot in biology. You'll have an enzyme and another enzyme that are part of the same process, and often these two will interact together so that they can be made and they can be in the same place. They'll just, slot together. Just like that. Okay. When is this important? Yeah, a lot of times. One example is going to be in sickle cell anemia. There is a mutation, and if you do one of these 23andMe or Ancestry.com or whatever genome screens, if you're into that sort of thing, then you can, this is one of the things they would tell you, I think. And so there's a little tiny change. It's just one amino acid that makes the difference between sickle cell and not sickle cell. And it's this little glutamic acid here turning into this valine over here. So glutamic acid, remember, is an acid. So that's beyond polar. It's actually got a negative charge. And valine is hydrophobic. And so by changing the amino acid, you actually change this little region right up here. And you change it in such a way that it's got a little pocket now, because valine's a smaller amino acid than glutamic acid. And it's a hydrophobic pocket. And so the hydrophobic surface of another one will slot together, and you'll end up like this. Normally, hemoglobin works really well because it can float around, and each hemoglobin can kind of independently pick up CO2, drop off oxygen, drop off CO2, pick up oxygen, just like that. When they're all linked together, they're kind of all in the same place, and each one is less efficient at picking up oxygen. And so, yeah, that these sort of chains explain why you get the cells with this particular shape. And these chains also explain why in sickle cell anemia, the hemoglobin doesn't work quite as well. So, yeah, it's not deadly, anything like that. It's just, a thing that a lot of people have. Okay, how do proteins get their shape? They have a little bit of help from their friends. Oh my gosh, yeah. So one of these friends is a protein called the chaperone protein. And this particular one is called a chaperonin, and remember the IN ending means that it is a protein. And so the little unfolded protein is going to be fed inside. This is going to change shape, and it's in a really nice environment that helps the protein to fold up properly. And then when it's correctly folded, it gets to pop right back out, and the cap's going to come off again, and the protein's going to be released. And now it's folded up. Most proteins can fold up on their own without the help of a chaperone, but occasionally, if it gets a little bit too hot or a little bit too cold, you can end up with unfolded proteins, or if you get one that's a little bit damaged. And the chaperone will come in here, and it'll refold that thing so you can keep using it. Eventually, we recycle all of our proteins. But we don't want to recycle them too fast because then we can't use them while we're recycling them, breaking them down. All right, how do we know about protein structure? Just if you're curious about these things, which, yeah, I hope you are. Yeah. There are a lot of different methods. One of those is X-ray crystallography, where you actually get your protein together and you get so much of it and so little water that the protein will actually crystallize. Kind of like a salt crystal, but a crystal of pure protein. Really neat. And then once you have that, you can shoot X-rays at it from all different angles. And because all the atoms in all the molecules are lined up in a crystal, they're actually always in the same place. And so if the X-rays are going in a particular direction, they will scatter off, and you get these little spots all over the place. And yeah, those can be solved. You can figure out the protein structure. Another method is nuclear magnetic resonance, where you are basically causing certain bonds to resonate. Think like wobble back and forth, if you like. And yeah, they will resonate differently depending on what is around and what they're interacting with. They can, do a little more or a little less. And then the other method that is one that I've worked in is called electron microscopy. You can just take a picture of it with electrons, by throwing electrons at it. It's like if you had an invisible car and you wanted to see where it was by throwing rocks at it, which is a terrible thing to do with a car. But you could throw the rocks, and if the rock just passed over, you'd say, yeah, no car behind this. But wherever the rocks bounced off, you could say, oh, okay, there's probably a car under here. And yeah, you ding the heck out of your car, and the electrons beat the heck out of the sample. But yeah, you can get a picture of its shape if you throw enough of them at it. So here's a little X-ray crystallography setup. You're going to have your protein crystal here. You can have a little X-ray source, and labs that do this will sometimes have their own little home source. But when it's time to do the real protein structure and get real high-level information from it, they'll always take it to a big thing, a thing called a synchrotron, which is a giant circle. It's a kind of a circular atom smasher. And yeah, this is going to constantly supply X-rays, and you're going to be able to use these to do wonderful structures. And there's going to be all little tiny spots in here, and that's the diffraction pattern from the crystal. And then from that, you can take it and figure out what the structure is of the protein. And look what protein it is. It's one that we studied, RNA polymerase II. Do you remember what RNA polymerase II does? Yeah, it's the one that makes every messenger RNA. It copies DNA, and so here we've solved the crystal structure of this actually bound to DNA, so there's our DNA, and it's going to read it and make an RNA copy. And so there you can see the RNA copy that matches the DNA and that we're just making right here. That's RNA polymerase II, and they've kind of caught it in the act and sort of frozen it in this position, which is kind of neat. All right. And so, yeah, DNA and proteins are definitely things that we can look at to try to understand evolution, because as DNA changes, the DNA codes for proteins, and so the proteins will generally change slowly, because proteins are like machines, and you can't just change out a part of a machine and expect it to keep working. The test of this would be, imagine you have a car, and imagine you take the spark plugs out of your car. Now try and put any other piece in place of a spark plug. That's basically a mutation. Is it going to work? Is your car going to work? No, it's not going to work. Because you need exactly that piece in exactly that position at exactly that spark plug gap in order to get your combustion engine to work. So some parts of the protein you can change, though. So imagine you change the the cigarette lighter for a USB charger. Is that okay? Is your car still going to run? Yes, this is a mutation that that can be withstood. And so the things in a protein that can change generally will change over time. So mutations will build up and change will happen. The things that can't change will still change, but every time they change, those individuals will die. And so that's natural selection, basically, right there. Any bad changes are going to be weeded out, and you're only going to be left with the changes that are at least neutral to slightly positive. That's what you're going to get in nature. All right, thanks very much. That's the end of this lecture.